Hello and welcome to Science Tutor's YouTube channel. My name is Professor Dodge, and today I'm going to show you how to very easily distinguish the erythrocyte from the leukocyte and the platelets when looking under the microscope at a human blood sample. Okay, so let's get started with that. And when we're studying microscopy virtually, we tend to use a photomicrograph, a picture of a histology slide, and that's what we're looking at here. This is a typical human blood smear. The formed elements have been stained using a right stain, so we have contrast and we can distinguish one cell type from another. The most numerous cells on the slide here, the most numerous cells in the circulation, are the red blood cells. And they're very easy to identify for three simple reasons. One, they're small. Two, they lack a nucleus. There is no nucleus in a mature red blood cell. And three, the cytoplasm in the center of the red blood cell is so thin that it hardly picks up any of the chemical dye, and therefore it stains clear, and it almost looks as if there's a hole inside the cell. Of course, there isn't. It just looks that way. So super easy to distinguish these erythrocytes or red blood cells from the larger white blood cells, also called leukocytes. They're not only much larger than the red cells, but they're fewer. And lastly, the third thing we notice is that the white blood cell has a nucleus. We'll learn in another video that there are 5 different types of white blood cells, and the nuclei in each type looks a little bit different. You might have a nucleus with 5 or 4 segments like we see in these 2 white blood cells, or there may be just 1 or 2 segments. So we'll review that later. The other thing is the cytoplasm in the white blood cell may or may not have some granules that pick up some additional staining. Regardless of which type of white blood cell you're looking at, they always look different from the red cells, again, because they're much larger, and they have this funny shaped nucleus. So we now can distinguish the erythrocyte from the leukocyte. And lastly, we want to be able to identify the platelets in a human blood smear. And here they are. And these almost look like debris on the slide, and that's because platelets are not cells. Platelets are simply fragments of cells. They're really just pieces of plasma membrane that fuse together to form a little sac-like structure called a vesicle. And the contents inside those platelets or those vesicles will be used to initiate a clot formation. So those are the 3 types of formed elements that you can identify on a human blood smear. You have the numerous erythrocytes, the leukocytes, and then those little fragments called the platelets. So hopefully this will help you when you're looking under the microscope at a human blood smear slide. And if you learned from this little mini review and you'd like to learn some more, we hope you'll subscribe to our channel. But more importantly, we'll hope you'll check us out at our website or send us an email if you require some additional tutoring or information on any anatomy and physiology topic. Hope you've enjoyed this and hope to see you soon. Take care. Bye-bye.